we know now that each cell inside of an organism is going to contain an identical copy, apart from sex cells, an identical copy of DNA in the form of chromosomes. And this DNA is kind of the program or the recipe for how to create different things which the body of that organism requires, and also programs for different kind of characteristics. So we've got all of this DNA stored in cells, but what happens if we want to create more cells? What happens if cells are going to divide into more and more cells in order to grow or replenish muscle tissue, repair damage in our body and things like that? So firstly, we'll just have a look at if we have one cell here, like so, say this is a eukaryotic cell, so it's going to have four linear chromosomes like that. What's going to happen is, in order for that one cell to become two cells, we're going to need to see DNA replication occur. So what would happen is each of those chromosomes would actually double its DNA, so replicate its DNA like that, in order to have enough copies, so a double copy, that we could then see those doubled chromosomes divide into two in order to create two identical cells. If we look at a prokaryotic cell, obviously we would not have a linear chromosomes, we would have a circular one, and then through DNA replication, that circular chromosome would become two circular chromosomes, which could then give rise to two identical cells. So in order for cells to be able to divide, in order for us to grow, in order for us to repair broken cells, damaged tissue in our body, etc., our DNA needs to be able to replicate first, because every cell in our body needs to be able to have a copy of that DNA. Now, if we go down to this diagram here, this is going to give us a kind of detailed run through of how that process occurs. So we're starting off with a chromosome. So initially our chromosomes would all be bundled together in this tangle of kind of spaghetti almost, in this tangle of spaghetti called chromatin. Now this chromatin would condense into its individual chromosomes for this process to occur. We've got our double helix, which you can see here. So we've got our DNA double helix. In order for DNA replication to occur, we need that double helix to unwind so that those two strands can then become two sets of two new strands. So four new strands, I guess, overall, two new double helixes. The first step in that unwinding of the DNA happens because of something called helicase. I might just draw a number 1 there for helicase. This will be step number 1. Helicase, which is an enzyme. So enzymes are things which can have particular roles and functions within cells. Helicase is an enzyme which unwinds the DNA double helix. So helicase helps to expose the bases to the rest of the cell. So we see the DNA double helix being unwound by helicase. So the two separate strands are opened and we get the exposing of bases. The next step, what will happen is nucleotides will base pair to expose bases. So if we look up at our diagram there, we can kind of see, we can see our free nucleotides there, and we can see a little bit of this happening in this section here, I suppose. Pop a little step 2 there. So you can tell by those free nucleotides there, we've got some different colors. Our green, our yellow, and our red are our bases, and then they've got this little blue square attached to them showing that that part of the nucleotide is there as well, so sugar phosphate backbone. So these will line up. These free nucleotides will be floating around in the inside of the cell, the internal environment, and when we get helicase unraveling the DNA, or unzipping the DNA, and bases are exposed on each strand, then free nucleotides will come along and pair via hydrogen bonds to the particular base that they want to pair with. Step number 3 involves another enzyme called DNA polymerase. Now what DNA polymerase is going to do is it's going to help attach those bases. DNA polymerase attaches the bases to the original strand. And finally, what will occur in step 4 is the new helix will wind up, I guess we could say. Or each new helix, rather than that, let's say each new helix, because we have now got 2. So each of these new helixes will wind up. We can see that sort of occurring there. Once DNA polymerase is connected to the bases, then they will twist back together into a double helix. A couple more things to note is that what we have in DNA is the 2 strands that make up DNA are not necessarily made equal. We have a template strand, and we have what is usually referred to as a complementary strand. I'm going to write that again, it's a bit messy. A complementary strand. So when they divide, one of the old strands from the original DNA in the newly created double helixes is going to be a template strand, and then in the other one it's going to be the complementary strand. Now the template strand is what actually contains our genetic material. The complementary one is just kind of there making up the double helix. Now, another thing to note is this process is also referred to as semi-conservative. And the reason why we say this as being semi-conservative is each of the new double helixes formed, each of the new segments of DNA formed is going to contain one of the original strands of DNA from our starting double helix and one newly synthesized strand from free nucleotides. So whether the new DNA contains an original template strand and a new complementary strand, or the other way around, it is said to be semi-conservative process because one comes from our original DNA, so one is old and one is new, one is newly synthesized. So I guess those are the main 4 steps to DNA replication, which are necessary in order for us to be able to pass on genetic material from cell to cell, and then eventually, I guess, organism to progeny.